Let's look at the composition of chromosome. Chromosomes are composed of DNA and protein. Most are about 40% DNA and 60% protein. Significant amount of RNA is also associated with chromosomes because these are the sites of RNA synthesis. The DNA of the chromosome is one very long double-stranded fiber that extends unbroken through the entire length of the chromosome. A typical human chromosome contains about 140 million nucleotides in its DNA, and the amount of information one chromosome contains would fill about 280 painted books of 1,000 pages each if each nucleotide corresponds to a word, and each page had about 500 words on it. Furthermore, if the strand of DNA from a single chromosome were laid out in a straight line, it would about 5 cm, fitting into a small piece of nucleus is nature's marvel, and that only one of 46 chromosomes. In the cell, however, the DNA is coiled, allowing it to fit into a much smaller space that would otherwise be possible. How can this long DNA coil so tightly? If we gently disrupt a eukaryotic nucleus and examine the DNA under an electron microscope, we find that it resembles a string of beads. Every 200 nucleotides, the DNA duplex is coiled around a core of 8 histone proteins, forming a complex known as nucleosome. Unlike most proteins which have an overall negative charge, histones are positively charged due to the abundance of the basic amino acids arginine and lysine, and are thus strongly attracted to the negatively charged phosphate groups of the DNA. The histone cores that act as magnetic forms that promote and guide the coiling of the DNA. Further coiling occurs when the string of the nucleosomes wraps around into higher order coils called supercoils. Highly condensed portions of these chromatins are called heterochromatins. Some of these portions remains permanently condensed so the DNA is never expressed. The remainder of the chromosomes called euchromatin is condensed only during cell division. When compact packaging facilitates the movement of chromosomes, at all other times euchromatin is present in open configuration, and its gene can be 125 expressed. Way chromatin is packaged when the cell is not dividing is not well understood beyond the level of nucleosomes and is a topic of intensive research.